Okay, hello. This is a video that will explain the stages of meiosis for the standard level IB curriculum of biology. This isn't more for higher level, but because I don't know that syllabus, but I believe this will help you understand at least the basics of meiosis and the stages. That's what I'll mainly be focusing on. Okay, so if you don't know, you should know certain principles about meiosis or genetics in general as a unit before you watch this tutorial, which are listed on the screen right now. This will help you visualize what meiosis really is and give you a clear image of what I will be explaining in the near future. And I get most of my information from a website, BioNinja. It's a very good website. It has 90% of at least 90% of the syllabus for the biology standard level course. I'm not so sure about higher level, but yeah, this video is mainly for standard level. But I'm sure people from higher level can learn something from it at least. So once you know all this, you can come back to the video and we'll get started. Okay, so first I'm going to go through some certain basics that you should definitely know. Meiosis. So you must know what meiosis is to understand the stages. So is sorry, meiosis is based off of two division sequences, and genetic sequences about separation and cellular division. And mitosis, which is completely different, you should try not to confuse them, only has one of each. In meiosis, homologous chromosomes separate, leading to daughter cells that are not genetically identical, whereas mitosis, the daughter cells are genetically identical. And meiosis is preceded by interphase, in which this is where the DNA grows and prepares for cellular division. And it consists of two sister chromatids. And then there's a second growth phase called interkinesis. This is where the cell actually splits, and no DNA replication occurs in this stage. And the biggest difference I'd say between meiosis and mitosis that you should definitely know is that meiosis, what you're learning about now, there is genetic variation. Whereas mitosis, there's no genetic variation whatsoever. Mitosis, you produce two identical daughter cells. Where meiosis, you produce four genetically different cells. Okay, so you must also know chromosomes. These are thread-like structures of nucleic acids and proteins found in the nucleus in most living cells, in eukaryotes usually. And this is what carries genetic information in the form of genes. So as you see down here, you can see that what a chromosome consists of. This is all you need to know, to be honest. The centromere gene, and there's two sister chromatids, as you can see here. And and then you must know what loci is, where is the point of where the gene is located, the genetic information. And yeah, that's what you need for chromosomes. By the way, most of the information that I gain right now really isn't that hard to find. I actually just Googled what each one of these are, and they gave me pretty good definitions for the IB. Okay, now you must know what a nuclear membrane is. Nuclear membrane is a double lipid bilayer membrane. It surrounds the genetic material and keeps it in place and prevents certain unwanted molecules or bacteria to enter, and contains all genetic information. And it also surrounds the nucleolus in eukaryotic cells. Okay, homologous chromosomes. These are made up of chromosome pairs of approximately the same length. In a centromere position, that's where the two sister chromatids connect, and the staining pattern for genes with the same corresponding loci, so they have the same genes. One homologous chromosome is inherited from an organism's mother, and the other is inherited from the organism's father. So each chromatid contains different information because they come from different organisms. This is what leads to variation. And a pair of homologous chromosomes are known as bivalent, which is everything I'm saying now is very important to know because this will help you visualize and memorize the process of meiosis. Okay, spindle fibers. Spindle fibers form a protein structure that divides the genetic material in a cell. So in this case, it will divide the chromosome into two. It will separate the sister chromatids. See this green stuff? It connects to the center of the centromere of the chromosome, And then later I'll show you in the stages, as I explain the process of meiosis, how it splits them in two. Okay, so now we're going to start talking about the actual process of meiosis. So as mentioned before, meiosis is split up into two division parts. So division one, once you learn the first part, the second part will be very easy to remember. It's almost identical. Okay, so meiosis division one. This is the first meiotic division that occurs, and it's a reduction of division. It leads from diploid to haploid, creating an X. Yeah, if you should know these two terms also. In which homologous, and this happens when homologous chromosomes are separated. Okay, so first stage of meiosis is prophase. Prophase 1. Okay, this is where chromosomes are condensed, nuclear membrane dissolves, homologous chromosomes form bivalents, the two homologous chromosomes. And this is, you must memorize this. Crossing over occurs. This is vital for you to mention each time in the IB when it mentions something about prophase. This will certainly gain you a point. Without it, you'll probably lose a mark. As you can see here, the nuclear membrane dissolves, so it shows it's dissolving, and soon the chromosomes will spread out, as you'll see in the next stage. So metaphase, the nuclear membrane has completely dissolved, and the spindle fibers, these lines, they go to the opposite parts of the cell, and from opposing centronodes connect to the bivalents at the centromere, and align them along the middle of the cell, as you can see here. So the nuclear membrane that contained all the genetic material leaves, and then they align them along the middle of the cell. Next is Anaphase division 1, or you could just call it Anaphase 1. I just put division 1 to help you remind you. So in this part, spindle fibers contract and split the bivalent. Homologous chromosomes move to opposite poles of the cell. So here you can see that spindle fibers connect to the centromere of each cell, and then they pull them away from each other. So this splits the two sister chromatids away, and they bring them to opposite sides of the pole, as shown in the diagram below. So telophase division 1, this is the last stage before interkinesis, and of like the separation part, as I'll explain later. So chromosomes decondense, nuclear membrane may reform. But you don't have to remember that may. If you say nuclear membrane reforms, you won't lose any marks. Okay, so this is where actually syntokinesis, sorry I made a mistake before, syntokinesis occurs during telophase. And this is where the cell divides into two to form two haploid daughter cells, and they are not genetically identical. So just imagine a line going through the middle of the cell, because each chromosomes are at the opposite end of the cell. And then there's a middle that goes along the middle of the cell, and they separate, becoming two haploid daughter cells. You must remember they're haploid daughter cells because they're half the genetic material of the previous mother cell, I guess. So now we're going to go to meiosis division 2, or meiosis part 2. This is the second stage that separates sister chromatids. And you must remember, chromatids will not, or most likely will not, be identical due to crossing over or variation in prophase 1. Okay, now if you remember what I said earlier, most of this will be very similar. You just have to remember that there's two haploid cells this time. So, as like in prophase 1, chromosomes condense, nuclear membrane dissolves, centrosomes move to opposite poles, and they're perpendicular to before. So yeah, this is this line, nuclear membrane dissolves, and then they'll go, and yeah, as mentioned before, now we're going to go to metaphase division 2, or metaphase 2. Now here, spindle fibers from opposing centrosomes, attach to chromosomes at the centromere like usual, and align them along the cell equator, or the middle of the cell. That's just a fancy way of saying the middle of the cell, like always. This time they're in a just different axis. Okay, and now we're going to talk about anaphase division 2. So spindle fibers here contract and separate the sister chromatids, and now called chromosomes because they're part of their own cells. They used to be chromatids, but then now that they're part of their own cells, they're considered chromosomes themselves. And they move to opposite sides of the pole, as you can see in the diagram below. And this is just like the first stage. Just you have to remember is that there's two cells now, pretty much. And then telophase, chromosomes decondense, nuclear membrane reforms, cell divides, syntokinesis occurs, and then two daughter cells that are not genetically identical appear, and in the end, there's a total of 4 daughter cells that are genetically different. Okay, so now if this is the end of the tutorial, this is the final overview of what happens. This is a diagram on BioNinja. If you go visit it, which I do suggest because there's very good resources there that can get you at least 90% of the marks on the test, you'll see this where it shows the interphase. This is where the cell prepares for division, but that's not part of really meiosis. It occurs after meiosis, but it is vital to know. And then, which you can find on BioNinja, you can find everything pretty much on BioNinja. Then prophase 1 happens, metaphase, and drawing them is very vital. This is a skill that you must know to draw the entire stages, which may sound difficult now, but once you get it down, you'll probably be praying that this question comes up, that you have to draw the stages of meiosis, because there's a lot of marks, and if you memorize it, they're very easy. They could give you 8 marks. So I hope this this video was helpful in any matter, whether you're standard level or higher level biology. And thanks for watching.